morning, everyone. So my goal, and I think I have 30 minutes-ish, and my goal is just to give you a flavor of the science that we've been working on and a sense of what we might be able to do as the Hunt MI project moves into the next very, I think, large stage of its growth. So the goal of my research is to understand the biological basis of cardiovascular disease through genetics, and this requires expertise and a sort of merging. We sort of work at the forefront of these three fields, both statistics, genetics, and also bioinformatics. So this is a little bit of an overview of what we mean by genetic disease discovery. So we start with something like cases and controls for a particular disease. We look at the DNA in cases and we look at the DNA in controls. And ideally, we're going to look at genetic data from tens of thousands to hundreds of thousands of people, which you're all well aware are fully available within the impressive resource of the Hunt study. Typically, we're going to examine millions of different sites in the genomes of these people. And these genetic sites are going to vary between cases and controls, and we try to identify which regions of the genome are different between cases and controls, and therefore at least associated with disease. We can also ask questions like which of these genetic sites, these genetic changes, are associated with quantitative traits, such as cholesterol levels or blood lipid levels, which are a very key risk factor for cardiovascular disease. So a major challenge, just to keep in mind as we walk through the rest of the slides, is in discovery genetics, is we identify a region of the genome that's associated with a disease, but how do we pinpoint the functional gene? And this is sort of the poster child for a region associated with total cholesterol where we just can't tell which gene it is. So we've identified non-coding variants. They span about 600 kilobases, which is fairly large from a genetic perspective, encompasses about 25 genes. We don't really know which one of those genes is responsible for changing cholesterol levels in people. So this is a major challenge that we focus on as time goes on. So I do want to give you a little bit of the history of the Hunt MI project and how we came to develop what really is such a wonderful collaboration. So somewhere, that red dot's in the wrong spot, but somewhere around 2007-ish, I believe, Dr. Cristen met up with this guy, Dr. Michael Benke, who's in the front row, and came from the University of Michigan, and they sort of said, hey, what cool things can we do together with these wonderful resources? And Mike's deep involvement in statistical genetics. And they came up with an idea, and then some of us sort of sat around and said, well, how can we make this happen? So my suggestion, since I was newly hired as a faculty member into the Cardiovascular Center at the University of Michigan, was, hey, let's do some cardiovascular genetics. And Audgar said, that's great, I love genetics. And we met with some cardiologists who said, great, I love cardiology. And the rest is history. So our very first project, so again, as a newly minted faculty member in 2010, I wrote a grant to the National Institutes of Health in the US the goal was to look for rare variants that are associated with myocardial infarction and blood lipid levels. So that's where the first MI part came in the Hunt MI project. I think we've grown to incorporate enough additional phenotypes that maybe we can just pretend the MI stands for Michigan. But either way, it works out. So our plan with this proposal was to perform whole genome sequencing of around 2,000 samples, selected from the earliest onset cases of heart attack or myocardial infarction, and matched controls. And after that sequencing, we would impute all the variants into additional samples, and I'll talk about that a little bit more later. The thought behind selecting the earliest onset heart attack cases is that perhaps they have more genetic factors that we might identify and fewer environmental factors. The NIH study section that reviewed our grant, I would say, was cautiously optimistic. We were given a 3rd percentile, but the comments were quite reassuring. We were called an excellent group who've been leading the field. They suggested that the findings here will be important for guiding future efforts for discovery of rare variants and complex traits. and that sequencing analysis from the Hunt study should provide a benchmark for many other studies of coronary artery disease. So that study began in 2011, and it actually took us about 4 years to get the sequencing completed. So in the meantime, on our weekly phone calls, we're asking each other, what can we do next while we're getting all the sequencing done? And we said, well, let's focus on coding variants. So this is the 1% of the human genome that encodes all of our proteins. And this is has the most obvious immediate biological impact. So we said, let's focus on these specific 1% of the genome. We developed proposals that two of which, one was funded by the liaison committee of the Central Norway Regional Authority here, and also an NHLBI, this is the National Heart, Lung, and Blood Institute in the United States, resequencing and genotyping service. So between those two funding sources, we ended up with a chip-based approach where we annotated about 200,000 variants, most of which impact proteins directly, in about 10,000 samples. 3,000 of those samples were all the independent MI cases out of the Hunt study, as well as matched controls. And this is just a sort of overview of the design. So we compared genetic variants in MI cases to controls. And then for our most interesting findings, thanks to a very nice collaboration with the TRUMSO study, we could attempt to replicate the most interesting things in the TRUMSO study before publication, so we could be completely confident of our findings. So our combined sample for this MI study was a little over 10,000 samples. We didn't find necessarily a home run in trying to find variants for MI. It turns out that variants for MI are very, very difficult to find and require extremely large sample sizes. But we did find some really interesting findings for blood lipid levels that gives us some clues about how MI might be, how risk of MI might be coming about. So the little black dots are things that we knew from genome-wide studies going into this, and the red dots are things that we can identify from this focus on the coding variants. And what we found was within the coding region of the genome, we're more likely to have rare variants with a very large effect. So they're less frequent in the population, but they have a substantial impact on your blood cholesterol levels and your risk of heart attack. So back to this problem of this really wide region, it's really hard to tell which gene in that region is responsible for the association with blood lipid levels and cholesterol. And this region was initially discovered in 2008 while I was a postdoc in Mike Benke's lab in collaboration with Sate Catharason, who's on the scientific advisory board, and Gonzalo Abecasas, who you'll hear from next. And this middle panel here, I don't know if there's a pointer, this middle panel here shows the association results from our coding array. So this is our focus on coding changes within the genome. And you'll notice this TM6SF2 variant gives us the exact same level of significance as the original top hit. Turns out those two variants are almost perfectly correlated. So they travel together in every person that we study. And this TM6SF2 shows very strong association with cholesterol levels and myocardial infarction. So genetic association is not cause. It just tells us two things are tied together, but we wanted some more convincing data that this specific gene, TM6SF2, that no one had ever really looked at before 2014 when we discovered this result, we wanted to really nail down that this gene was responsible for impacting cholesterol levels and heart attack risk. So we collaborated with Eugene Chen, who's a faculty member in my division of cardiology at the University of Michigan. And he's an expert in developing mouse models for cardiovascular disease. And he said, oh, well, that's easy. We'll just disrupt this gene in mice and see what happens. So it sounds easy to us. I'm sure it's not that simple. But so using adeno-associated virus and tail vein injection, they both overexpressed the human form of TM6SF2, so he gave the mice extra copies of this gene to see what happens. And when he does that, he sees that both total cholesterol and triglyceride levels are increased. Those are the yellow bars. And then when we do the opposite thing and say, well, let's knock down the natural version of TM6SF2 in the mice, you see the opposite effect. So this is much more convincing evidence that this gene is specifically causally related to cholesterol levels and heart attacks. So this enabled us to publish through this collaboration, and as well as the functional follow-up, which is really critical, allowed us to publish in Nature Genetics in 2014 these findings, which were published back-to-back with another paper from a group in Texas led by Helen Hobbs, who found that, so we see one allele is associated with higher cholesterol and heart attack, and what her group sees is that the other allele is associated with fatty liver disease. So you can, it seems like you can sort of choose. Would you rather have your lipids circulating in your blood, piling up in your arteries, potentially giving you a heart attack, or would you rather have them clogged up in your liver, giving you fatty liver disease? So the mechanism how TM6SF2 is not completely worked out, but it's a very exciting region that's been a major impact in the research community. Now, I did mention that MI is a tougher disease to study. There just aren't very many of this, what we call low-hanging fruit. These rare variants of large effect just don't seem to be present for myocardial infarction. So we did publish a paper saying, you know, we did this, these kinds of findings are also really important for the scientific community, because if someone wanted to replicate the same experiment, we can say, you know, we had a look, this is what we see. So we published this paper saying that we didn't think there were any of these low-hanging fruit for MI. And through a collaboration that was mostly led by Sate Catheracin, we put together a consortium of, I can't remember how many studies are in here. It's probably on the order of 30 or 40 different studies who all did the same similar sort of experiment that we did. We put all our data together, and in sample sizes that are absolutely enormous, so 73,000 patients with coronary artery disease, 121,000 controls. So a much larger sample size than what we originally looked at. They found a total of 3 variants associated with coronary artery disease. So clearly, extremely large sample sizes would be needed for making novel discoveries for this sort of tough disease to study. So in a similar vein, we've contributed the data that we've generated to a number of different consortia. We've collaborated with many different groups. Some of these groups are doing statistical modeling, method development. Some of them are meta-analyses for a variety of different traits. But we're contributing this data in a very broad way to many different consortia who depend on this data to exist. Some of these consortia we also lead. So again, while we're still waiting for our sequencing to get done, we're also working on a number of different projects at the same time. So this is just an example of a project both Gonzalo and I worked on at the National Institutes of Health, funded by NHLBI. And the goal was to perform targeted sequencing of individuals with extreme lipid levels. So again, this idea that people with very, very high cholesterol and very, very low cholesterol might have more sort of genetic load. They might have more genetic variants of high effect. So we sequenced extremes for LDL cholesterol. We ended up with a sample size close to 3,300, which between the time of sort of 2011, 2013, when the study was a very large sample size for whole exome sequencing. And we discovered one novel gene associated with blood lipid levels called PNPLA5. It's a lipase. It's a strong biological candidate. We're still sort of working out the biology. But you can see in this, I don't think there's a pointer. You can see in the what we call the violin plots on the right that the individuals in pink with a loss of function or a missense variant in that gene have much higher LDL cholesterol levels on average than people who don't have a mutation in this gene. This work was done by Yuna, who was a graduate student at Michigan at the time. And then we also discovered 3 very well-known lipid genes where loss of function mutations cause a very severe phenotype in terms of cholesterol levels. So the LDL receptor mutations in this gene cause familial hypercholesterolemia, and you can see that individuals with a loss of function of this gene here in this column have extremely high LDL cholesterol levels. If you have a single amino acid change in the gene, again, you have higher levels than people who don't have a mutation. The opposite is true for these two other genes. So the apolipoprotein B, if you have a rare variant in this gene, you have very, very low LDL cholesterol levels, and you're also protected from getting heart attack. And then this is a fascinating gene that was first published in 2005, and I think it was in 2015, 3 drugs came on the market that inhibit PCSK9 very, very effectively. But natural people who have a natural sort of broken copy of PCSK9, which is this protein is involved in recycling the LDL receptor. If you have a naturally broken copy of the gene, you have naturally low LDL cholesterol levels, and you're protected from getting heart attack. So this is a beautiful example of really what we hope to find from genetics. A gene that, when broken, protects you from disease, and that we can, I don't mind walking around, keeps me awake. But then when we target this gene with a drug, we can reproduce that in everybody. So that's really the gold standard of what we're hoping to look for. I better go faster, talking too much. So another project in the lab was to repeat these experiments in individuals from China, because we see different genetic variants from individuals in China. So this is a different collaboration that we work on at Peking University in Beijing. So we also wanted to repeat this study in as large a sample size as we possibly could. So through the Global Lipids Genetics Consortium, which I help lead together with Mike and Cristen and Gonzalo, we put together a sample size of about 300,000 individuals. Through that study, again, of the coding region of the genome, we've identified 35 novel loci and associated with lipids, and 40 novel ones that are in the other parts of the genome. And we're writing this up. I just saw another draft come around last night. So I'm going to skip this in the interest of time. Another area that my lab's very interested in is functional interpretation of what the non-coding parts of the genome are doing. We know that there's important regulatory regions within our DNA. We're not necessarily that good yet at pinpointing where they are. But one of the first things we can ask is, for what we understand about these regulatory regions, do they overlap with the regions that we find associated with lipid levels or with heart attack? And so we developed a statistical tool that allows us to ask that exact question. Do we see overlap more often than you expect by chance between variants associated with disease and variants that we know fall in regulatory regions of the genome? So my graduate student, Ellen Schmidt, developed some software that's now been published in the journal Bioinformatics that allows us to identify correlation between regulatory regions, such as strong promoters or enhancers, inappropriate cell types, to help us understand the mechanism by which non-coding variants might be impacting disease. So in the meantime, our sequencing of 2,200 individuals was completed. Of course, it wasn't that simple, and we were doing analysis all along the way. But so around 2014, we had some data that we could start to look at. So we, of course, immediately said, well, let's test for association with MI and lipids for everything that we can think of, both all these single genetic changes, whole genes, structural variants. But remember, I showed you that example of a paper with 70,000 MI coronary artery disease cases, 120,000 controls. They found 3 hits. So keep that in mind. We're studying 2,000 people here. There probably isn't any low-hanging fruit. We didn't really find anything. But that's okay. We know our sample size was too small. We need to move forward. So we got together to sort of regroup and figure out what's the best thing to do with this data, and where should we go from here. Cristen, I think maybe you hadn't quite had your coffee yet, or perhaps you're praying. But but we had to think about what should we. Yes, saying grace. So I think we thought about what to do next. And Gonzalo, who's a big thinker, said, we should just genotype the whole hunt sample. And my memory of what happened next was Cristen said something like, I think our research subjects would really like that idea. And Odger said, that sounds like some really cool science. But of course, he was actually thinking, I wonder how much IRB paperwork that might be. So the experiment we designed moving forward with this data was, well, let's do GWAS in all of our hunt samples. And what that means is that we'll have a sort of slightly sparse view of the genetic information in those individuals. We sort of genotype markers as sort of signposts within the DNA. But then we can take advantage of these 2,200 fully sequenced individuals. And we can actually impute the genetic information that we see here into these individuals. So it might be that this person might actually be a second or third cousin for someone that we've sequenced. And we can actually figure out what the genotypes should be at the places that we're missing. So it works something like this. Based on what we observe, we can fill in the rest of the genotypes by picking a best match from our sequenced panel. So we fill in these genotypes in all of the 68,000 hunt samples that now have genome-wide markers genotyped. And we end up with a pretty good guess of the genetic variation across all the markers of the genome. So our newest hunt project that we submitted again to NIH as an R01, we thought we'd focus on understanding the biology of atrial fibrillation. There hasn't been as much research done in this area as there is for coronary artery disease. So the biggest genome-wide association study is in the few thousands instead of the few hundred thousands. So it hasn't been looked at as carefully. So what we said we'd do is we'd do whole genome sequencing of 2,000 samples. And here because the technology's changed, we can do deeper sequencing than we did before, with imputation into the rest of the Hunt MI samples that have now been genotyped. And also we partnered with a group that develops functional experiments to understand the mechanism. And so this group in Michigan has developed from human-induced pluripotent stem cells. They differentiate these stem cells into cardiomyocytes that in a 96-well plate, these cells actually beat in the dish. So what you can do is you can introduce genetic variants that we find in the Hunt study into these different cells and see how that impacts how these cells beat in the dish. It's pretty unbelievable. So actually, again, we sent it to NIH. They were, again, cautiously optimistic. They came back to us with a second percentile, which is good. And they said that the large and well-phenotyped cohort available for analysis make the chance of success very high. And they also called us, I'm slightly embarrassed to put this up here, but an outstanding team of investigators. And we'll just move on. So I want to give you a little overview of just the scale of how things have changed only in the last 5 years. So when Mike and Cristen first got together, they did some metabochip genotyping. I think it was a couple thousand samples. And that's actually 400 million genotypes, which has moved on over time. So our exome chip study that I mentioned was funded by the liaison. Yeah. But 2 billion genotypes. Our whole genome sequencing of 2,000 samples that took us 4 years to complete is about 44 billion genotypes. The Hunt all-in GWAS, 68,000 samples at about 500,000 markers is about 40 billion genotypes. And now that we can impute all those variants into the whole Hunt sample, we're talking in the trillions of genotypes. So you can see how the scale of the data that we've been able to generate over the last 5 years has really changed. So last week, we discovered a new gene for atrial fibrillation. It turns out there's about 6,000 some odd cases in the Hunt study that we've genotyped that have atrial fibrillation that at this point we can diagnose with ICD codes. We're not, we're not completely confident since it's only been a week and we have to kind of kick the tires and we have to confirm that this is truly a new association, but it looks very promising at this time. So we immediately went to our collaborators. This is not data based on this gene, but this is just what the kinds of things that they can do. So these are the beating cardiomyocytes in well format in the dish. Then they can take each well and measure metrics of the electrophysiology of those cells. So for example, again, this is just sort of a cartoon, but you could disrupt the gene in several of these cells and then quantify how that changes calcium influx and other measures of the electrophysiology of this cell. So they're working on it, and maybe next time we get together, we'll give you an update of how that turns out. So what do I envision as the next stage for our HuntMI collaboration? We'd like to continue to make new scientific discoveries related to cardiovascular disease as well as human health. It's important to understand the biological implications and the mechanism underlying these genetic discoveries. And I think the optimal approach is by partnering with experts in molecular biology who are world leading at developing these kinds of experiments. We'd like to integrate human genetics with other omics and other things that we can understand, biomarkers and epigenetics, to develop a more complete view of what is obviously a highly complex system, as well as developing prediction tools and bioinformatics methods for interpreting the function of genetic variants, particularly in the non-coding part of the genome, which is where most of these variants actually lie. And what are the implications for precision medicine? So those are some sort of topics that we're hoping to look at moving forward. This is my little cartoon of how our genetic discoveries sort of work in my mind. We start with people in a biobank. We develop these tools. We do some sequencing. We have a pipeline for the analysis. We're very careful about what comes out of that analysis to be completely confident of the findings. And then we decide, based on biology and other things, which of these interesting findings we should then invest in million-dollar experiments by developing animal models to further understand the mechanism. That hopefully leads to new therapeutics, which we can then move into clinical trials and develop new treatments for our research subjects and patients. So this has been a big group, a big team effort. Many people from the NTNU side, as well as from the Michigan side, and it's been an absolutely wonderful collaboration. Thank you.